Suresh here, your Games Academy faculty for Biochemistry. As a part of Fmge case-based test series for the session June 2023, I have come up with another case-based question. So let us read the question. A 3-year-old fair boy was brought to the hospital with complaints of delayed developmental milestones, mental retardation, seizures. He had light-colored hair, skin and eyes. His urine had mousy odor. Blood phenylalanine levels were more than 30 milligrams per deciliter, and normal supposed to be 1 to 2 milligrams per deciliter. So with this itself clear cut, you can have idea. Blood phenylalanine levels are more than the normal, almost 10-fold threshold, like 30 milligrams per deciliter, which is supposed to be 1 to 2 milligrams. At the same time, the urine, that boy's urine smelling like mousy odor, right? So that means it is indicating that this case is related to abnormality in phenylalanine metabolism. So it is one of inborn errors of phenylalanine metabolism. So here there is a delayed developmental milestones, light colored hair, skin and eyes, right? So it is indicating albinism, right? And delayed developmental milestones and mental retardation seizures also observed. So what are the probable, what is the probable diagnosis? What is the cause of the disorder and its biochemical basis, clinical features, diagnostic tests to confirm the disorder, how the condition can be managed, and why early diagnosis and treatment is essential in case of this abnormality. Right, we'll see the explanation. So if you see the phenylalanine metabolism in our body, being essential amino acid, phenylalanine is supposed to be supplied through the diet. Right, once we have consumed the phenylalanine, this phenylalanine in the body, in the cells, it will be converted into tyrosine with the help of an enzyme, phenylalanine hydroxylase. So this is the key enzyme which converts phenylalanine into tyrosine. That is the reason tyrosine is non-essential, as it synthesizes from phenylalanine in the body cells. Right, it is an oxygen-dependent enzyme, and the coenzyme which required, that is tetrahydrobiopterin. Here, this tetrahydrobiopterin converts to dihydrobiopterin. Again, this dihydrobiopterin regenerated to tetrahydrobiopterin by using the enzyme dihydropteridine reductase. Here, the main use of NADPH. NADPH will be supplying the reducing credentials, right? We are all aware what are the important substances you will be getting from phenylalanine. And once phenylalanine converted to tyrosine, tyrosine is useful for the synthesis of melanin. Okay, skin pigment, color pigment for skin and hair. Norepinephrine, they are all neurotransmitters. Dopamine, so which is excitatory neurotransmitter. Thyroxine, which is the main thing for thyroid hormone, right? So all these things, important substances will be synthesizing from phenylalanine, from tyrosine, from phenylalanine via tyrosine. So first thing is, whatever the products of tyrosine are same for phenylalanine, but the problem is phenylalanine first has to convert to tyrosine. As tyrosine is not converted, the biological important products will not be produced. So it is the most common inborn error of amino acid metabolism, that is phenylketonuria. Phenylketonuria is of several subtypes are there. If you see, I will explain that in next slide. So it is again autosomal recessive disorder similar to alkaptonuria. Its incidence is 1 in 10,000 births. If you see alkaptonuria, that is 1 in 250,000 births. Here phenylalanine hydroxylase gene spans 170 kb located in chromosome 12q 23.2. The majority 50% missense mutation, 10% splice site mutations. So the types, if you see, classic phenylketonuria because of complete absence of phenylalanine hydroxylase activity. The second is persistent hyperphenylalaninemia, that is low phenylalanine hydroxylase enzyme activity. Third, transient mild hyperphenylalanemia because of mutations, maturation or delay of phenylalanine hydroxylase. Dihydropteridine reductase deficiency, which is useful in regeneration of dihydrobiopterin to tetrahydrobiopterin. As there is no tetrahydrobiopterin, there is again inactivity or failure of working of phenylalanine hydroxylase enzyme. It is because of deficiency of absence of dihydropteridine reductase deficiency. Abnormal dihydrobiopterin function, dihydropterin synthesis itself defect. So based on these, you can classify the type of phenylketonuria. So what is the theme? Like seizures, mental retardations, delay in development of milestones. Why? Because Delayed milestones, you can see small head microcephaly, mental retardation, failure to walk, to talk, seizures, psychosis, tremors, and failures to grow. Because melanin, because light colored skin and eyes, you can see because of absence of melanin. So that's the thing. Increased phenyl and its metabolites toxic to brain. Phenyl and its metabolite compete with tryptophan and tyrosine in their metabolism, impairing synthesis of neurotransmitter serotonin. Obviously, serotonin is also produced, will not also not produced. So as what happens? So phenylalanine increases in the circulation. It leads to high levels of phenylalanine and it starts excreting in the urine, leads to phenylketonuria. So why the urine or sweat of the boy smells like mousy odor? Why? Because high levels of phenylalanine, they take alternative pathway. Okay, that is phenylalanine will be converted to phenylpyruvate and phenylpyruvate to phenylactate and phenylacetate and finally phenylacetyl glutamine. Understand? So this phenyl acetate is the main reason for mousy odor. Okay, the same smell when you enter into a biochemistry lab, that pungent odor what you get because of the acetic acid leakage or something, the same the smell of urine of phenylketonuria patient is also like mousy odor. Okay, because of high levels of phenyl acetate in the urine. Okay, and high pigmentation, yes, because of absence of melanin, hypopigmentation, lighter skin, hair color, blue eyes and eczema. So what are the diagnostic tests for phenylketonuria? There are like ferrichloride test again, like similar to alkaptonuria. Go3 test is there. It is a microbiological test, bacterial inhibition assay, heel prick method. Okay, so from the heel of the baby, they will prick and they will collect the blood and they will put it in a bacterial culture. So Bacillus subtilis is the bacteria. If there is a like what to say, if high levels of phenylalanine is there, there is a drug is already in use. So this phenylalanine inhibits the drug and allows the growth of what to say, allows the growth of the Bacillus subtilis. If there is low levels of phenylalanine, there is no inhibitor to the drug, so it decreases or it suppresses the bacterial culture. Okay, that is the thing. Blood phenylalanine level normal, as I said, in the cases will be discussed 1 to 2 milligrams per deciliter. Increased level up to 20 to 70 milligrams per deciliter. Newborn screening, tandem mass spectrophotometry is a gold standard to identify phenylketonuria in 24 to 72 hours after birth. DNA probes, cloning of genome for human phenylalanine hydroxylase. That is one more advanced technique. So to talk about malignant hyperphenylaninemia, that means dihydropterin reductase deficiency, which regenerates tetrahydrobiopterin. Dihydrobiopterin can be converted to tetrahydrobiopterin. Okay. So phenylalanine converted to tyrosine with the help of tetrahydrobiopterin. Again, tyrosine will be converted to L-dopa with the help of same tetrahydrobiopterin. Tryptophan will be converted into 5-hydroxytryptamine, like tryptophan hydroxylase enzyme. That is again it is dependent on the tetrahydrobiopterin. Arginine converted to citrulline in form of nitric oxide. Again, it is a tetrahydrobiopterin. So this is the importance of tetrahydrobiopterin and regeneration by the enzyme dihydropteridine reductase. So this tyrosine useful in making of melanin, dopamine, norepinephrine, epinephrine, thyroxine. Whereas L-dopa is for dopamine, prolactin increased levels, norepinephrine, epinephrine. Hydroxytryptamine is useful in making of serotonin and melatonin. And nitric oxide, vasoconstrictor, vasodilator, potent vasodilator. So treatment, because here the defect is phenylalanine hydroxylase. So instead of supplying the phenylalanine, if you provide tyrosine in diet, then tyrosine will become conditionally essential. In case of, so again is one more question you can make out. Example for conditional essential amino acid. Conditional essential here, the condition is phenylketonuria. So as phenylalanine is not converting tyrosine, tyrosine will become essential. Condition is making tyrosine essential. Otherwise, tyrosine is non-essential. So low phenylalanine diet supplement with tyrosine. Start within 3 weeks of birth to prevent irreversible brain damage. Long chain polyunsaturated fatty acids are prescribed. Glycomacropeptide protein derived from cheese whey. High protein diet is supposed to be avoided. Aspartame also we have to avoid in the diet. Defect in dihydropiranidine reductase, you can say tetrahydrobiopterin to be provided, sapropterin dihydrochloride company. L3,4-dihydroxy phenylalanine L-dope also supposed to be provided. 5-hydroxytryptophan, which is a source of serotonin, is also supposed to be provided. The first and only enzyme therapy approved for adults in living with phenylketonuria, that is Fmge. Pegvilicia is a gylated phenylalanine ammonia lyase, phenylalanine to ammonia and trans-cinnamic acid. This is the one which converts high levels of phenylalanine to ammonia and trans-cinnamic acid to excrete in the urine. So gene therapy clinical trial testing is under the safety and the effectiveness of adenovirus vector mediated gene therapy for phenylketonuria. So maternal hyperphenylalemia, so women with phenylketonuria can have normal chain. So phenylalanine crosses placenta, birth defects like mental retardation. Planning for pregnancy, you are supposed to check your phenylalanine levels. Proper education and communication about phenylketonuria of women and relatives. Measure phenylalanine levels before conception. Phenyl level throughout the pregnancy and diet management. So these are all the biochemical basis, enzyme defects, diagnosis and treatment, management of phenylketonuria. So that's all about it. Thanks for watching. Thank you.